Hi, I'm Tian Cai. Advances in biomedicine is always fueled by the exploration into uncharted territory of human biology. In this video, I will present our research on one of such challenges. Structure-enhanced deep meta-learning predicts uncharted chemical protein interaction on a genome scale. This is a work in collaboration with my advisor, Lei Jie. First, let me start ask a question. What if we can predict genome-wise chemical protein interaction? If that's doable, more undruggable targets will become druggable. And among the druggable targets, currently only 10% have approved drugs. If we can do genome-wise chemical protein interaction, the bias in functional characterization of human protein will be lifted. And for polypharmacology will be enhanced for multi-genetic disease, such as Alzheimer's disease, and genome-wise off-target prediction will be efficient, which will greatly increase the success rate of drug discovery. Given this motivation, then the question is, what are actually the obstacle to do this? In the age of big data, known chemical protein interaction, CPI data, is still very limited compared to the uncharted dark space. Here on the plot, each bubble is representing a protein family, a PFAM. Then we calculate the ratio of proteins that have at least one known small molecule ligand to this PFAM. Then you might have the impression that in PFAM1, we have a lot of data. Yes, but even for PFAM1, the illuminated ratio is still less than 1%. Meaning most of the protein in PFAM1 doesn't have a known binding ligand. And all those data, all those bubbles put in together is only 10% of all the protein families. There are vast, dark chemical genomic space out there for us to illuminate. That's the uncharted dark space. So what current methods do we have to generalize to the uncharted dark space? You might first want to say, can we get enough structure for the protein? As you know, it's not always easy and there will be prohibitively high cost at a genome-wise. We very excitedly have AlphaFold2 recently, giving you all the structure of any protein, but the problem is it doesn't have a high enough accuracy in terms of CPI prediction. We did some tests on dark protein. The test score for AlphaFold2 based docking is only around 0.5, which is just a random guess. And other sequence-based deep learning CPI protein prediction model, the problems with them, the generation problem. We did some tests by splitting the test set differently. We see the AC score dropped from 0.9 to 0.4 or even worse for those standardized methods. We proposed ProtoCG, the first solution that can solve the problem. From a biology perspective, it's a framework following sequence structure function paradigm. And CPI in the end is to predict the function of a protein. And hopefully all the information of function can be encapsulated by the sequence, encoded in the sequence. As AlphaFold2 has been doing, you predict sequence, predict the structure just based on sequence. However, in terms of CPI prediction, given the same sequence, protein can have different conformation binding to chemical at different binding site and have a slightly different change of structure. This is critical for CPI prediction. That means for more than what AlphaFold2 is doing, you need to incorporate not only sequence information, but also structure information at the binding site. That's what we do, bringing sequence structure information to the final function prediction. From accuracy perspective, ProtoCG provides a system-level control of auto-distribution generation at all the major building blocks of a learning system. ProtoCG has three major components: stepwise sequence structure function transfer learning, and auto-class meta-learning as an optimizing algorithm, and third, a stress model selection, which will be the evaluation logic to select best model instance. Please read our paper for more details. In this short video, we will just present the outline of the structure of our algorithm and also the major result that we want to demonstrate that PautoCG can be used to illuminate a dark space. We compare PautoCG with two baselines. DICI is a state-of-the-art deep learning method for orphan receptor API prediction, and PLD sign is a AlphaFold 2-based docking method with a state-of-the-art graph neural network to predict CPI. Compared to these two baselines, PautoCG not only have a major improvement on PRUC score, which is 18% improvement, it also, most importantly, have a very stable deployment gap at around 0, meaning that when you generalize your model with your training data to the dark space with auto distribution point families, you can expect the same test score as you tested with your known data. This is a major obstacle, stop most of the current deep learning methods. For example, DICI, we see a major AUC score dropped by 0.3, which means it can not be used to generalize into the dark space. So that will be just a brief introduction. Feel free to reach out to us, and we look forward to talk to you next week at Wisconsin. Thank you.